Hi, my name is Kevin Chen, and I am a third year MD PhD candidate in Dr. Darrell Khan's lab and the Center for Regenerative Medicine, or the CREM, over here at Boston Medical Center. Um, Today, I'm excited to talk to you about this project in which we've transplanted these mouse basal cells into a mouse model of cystic fibrosis. And um, I'm not going to talk too much about the pathobiology of cystic fibrosis itself, or some of these small molecule modulators that are really effective and have really revolutionized care. Instead, I'm really talking about this alternative avenue of therapy for cystic fibrosis, which is cell therapy to treat um, this kind of monogenic airway disease. And the reason why cell therapy could be a viable alternative is because there are patients who suffer severe side effects from the small molecules or are refractory or don't respond to those modulators at all. And so this wheel cell therapy has been the dream for a lot of us over here in regenerative medicine, because we can imagine this in a future where a patient with a genetic disease comes into the clinic and we are able to take a somatic cell from this patient, reprogram them into an iPSC or induced pluripotent stem cell that has the potential to give rise to any cell type of interest. We can correct their iPSCs to correct for any of their underlying genetic mutations, and then following directed differentiation protocols, generate them into a specific cell type of interest by really marching them along very specific developmentally guided milestones. And with the cell type of interest generated, we can then transplant these cells back into the same patient. So, given that this is the goal, where are we today? At least for something like monogenic airway disease like cystic fibrosis, right now we are modeling a preclinical set of transplantations. So, here in this project, we've isolated healthy basal cells from a wild type mouse and transplanted them into a mouse that has no CFTR function. So we've been talking about basal cells, but we're interested in using the basal cell because for many years now, basal cells have been established to be the resident stem cell of the airway. That means they give rise to all of the cell types in our pseudostratified layer in our airways, including these rare cell types, including the ionocyte, which I'll point out as having the highest levels of CFTR. And so, given that we have this cell type to transplant, um, we've really made a lot of advances in understanding how to develop an actual engraftment model. Last year, our previous MD PhD student published a paper in which we showed that we can transplant and engraft these primary or pluripotent stem cell derived basal cells into syngeneic mouse recipients following detergent conditioning of the airway, in which we slough off the luminal epithelium. If we do that and then give the cells after conditioning, then explant a trachea 4 to 5 weeks later, we might get an image that looks like this, which is a trachea that's been filleted open, so it lies open like a book. And here you can see green cells covering the entire luminal surface of this trachea. And so these green donor-derived cells that cover the surface, and when we phenotype them, we can see that they reconstitute really the expected repertoire of the airway cell types, including the acetylated tubulin positive ciliated cells you can see in the top panel, the 1A1 positive secretory cells in the middle panel, and the keratin 5 positive basal cells as well. So given that we have this engraftment platform, how do we actually develop a preclinical in vivo model of cell therapy for CF? Well, this is in collaboration with our colleagues at Yale, Emmanuel Bouchia, Marie Egan, who are experts in physiologic readouts of CF in a mouse. And so the mice that they have down there is a CFTR null mouse that is intestine corrected. And this mice, while they don't phenocopy human airway disease, in which they don't have bronchiectasis or recurrent bacterial infections, they do display very abnormal CFTR dependent current with a subtle airway phenotype that has mucus impaction. And so our goal here is to transplant our wild-type basal cells into these null mice to see if we can really rescue this abnormal CFTR-dependent current as a real first proof of principle for testing epithelial stem cell transplantation. Um, And so for disease rescue, here's our experimental scheme. We took primary basal cells from the trachea of a UBC GFP mouse, in which all of their cells are green, expanded these cells ex vivo, and then transplanted them into the CFTR null mice down at Yale. And so our hypothesis is if we were to slough off the epithelium of a CFTR null mice, so this epithelium that doesn't have CFTR function, and then we repopulate it with epithelium regenerated from these exogenous healthy basal cells, we might expect that this presumably healthy airway epithelium should reconstitute the CFTR dependent current. And so going into this, we transplanted these mice, and 6 weeks later, when we look at the trachea, we saw an image that looked like this. So we got successful transplantation. We have donor-derived cells covering the luminal surface of these CFTR null mouse transplant recipients. And when we look at the histologic phenotype of what our green cells have reconstituted, you can see in this immunofluorescence image, they've reconstituted our airway lineages, as well as these BSND positive green positive ionocytes. So these are rare cell types and imaged with help from our friends at the Cystic Fibrosis Foundation. So on taking these trachea and then imaging them, we can then digest them to see if we really reconstituted the CFTR message in these CFTR null mice. And so when we digest these trachea, extract RNA, and then make cDNA and run RT-qPCR, you can see on this gel in our CFTR null transplant recipients over on the right, you see we've reconstituted message, the CFTR message in virtually all of the recipients, as compared to a CFTR null naive mouse, third from the left, which has no endogenous CFTR message nor GFP. So how do we understand what we've transplanted a little more deeply? We can look at single-cell RNA sequencing of our transplant recipients. And so for this single-cell sequencing experiment, we really sought to understand what our donor-derived cells gave rise to, and what are the cell types and what are the gene programs that these cells are expressing. And so for this single-cell RNA sequencing experiment, we sequenced 3 mice, one a CFTR null naive mouse, along with 2 wild-type cell transplant recipients. And when we digested their tracheas and sorted for green and non-green cells, we obtained a dimensionally reduced UMAP projection here of all the cells. Here, this is an epithelium-enriched UMAP where you can see really all appearance of all 3 mouse cell populations, and we see GFP transcripts highly expressed in all of our donor-derived cells. When we label and annotate our cell clusters, we can see that our donor-derived cells really reconstitute all of the expected airway cell lineages in roughly the same proportions to the endogenous their endogenous counterparts. Finally, if we look at this very busy dot plot, the main takeaway here is if we look at we compared our donor-derived cells to their endogenous counterparts compared to a naive mouse, and we looked across all of the airway cell lineages and their canonical markers, we don't really see any overt gene expression differences between, for instance, a donor-derived basal cell with a endogenous basal cell with the naive basal cell. So the real question now is, given that we kind of know what are the cells that we've transplanted, and we know that the epithelium looks much like a normal mouse epithelium, does this new regenerated epithelium rescue any functional electrophysiology defects? And so upon explanting these trachea, our collaborators down at Yale are able to put them into a Using's chamber analysis assay where when you suspend the trachea between 2 fluid-filled chambers, you can add small molecules like forskolin, which activates adenyl cyclates, which then increases the expression of all cyclic AMP dependent ion channels. And you can detect this current generated by this addition of this molecule in the readout of a short circuit current jump. So when you add forskolin and you activate cyclic AMP dependent channels, you get a boost in the short circuit current. To really quantify the contribution of the CFTR channel specifically to this boost in short circuit current, you can then add a CFTR channel specific inhibitor, such as CFTR inhibitor 172, which will then decrease the short circuit current proportional to the contribution of the CFTR channel in that sample. And so the red line on this graph really represents the expected tracing of a CFTR replete cell. If we look at our mice, when we look at the green bar, which are the CFTR null mice that have been transplanted with wild type cells, you can really see that they reconstitute the forskolin stimulated current, short circuit current, much to the same levels as a control, which are wild type or heterozygous litter mates that have not had any experimentation done to them. And also similar to heterozygous animals has been transplanted with wild type cells. So we are reconstituting this forskolin stimulated current. And if we ask, what is the contribution of CFTR in these transplant recipients? Well, when you add the CFTR inhibitor, you can see that in our CFTR null animals transplanted with wild type cells in the green bar, that contribution or the magnitude of the current change is equivalent or not statistically different from a control sample and statistically significantly greater than any CFTR null naive mice. So this really shows that we can reconstitute functionally a CFTR dependent current by transplanting CFTR null animals with wild type basal cells and then reconstitutes fully healthy epithelium that rescues this kind of electrophysiologic defect. And so hopefully this is one step along the way to the clinic in which we have a mouse model in which transplanting their endogenous stem cells really reconstitutes electrophysiologic function. Along the way to the clinic, there will have to be larger animals and comparative physiology studies. But as a preclinical platform, this is really the first example in which we've been able to show disease rescue by transplanting an epithelial stem cell and engrafting it. So thank you for listening. And work like this really can't be done without the help of many, many talented individuals. And so they're listed here on this slide. And I'm happy to take any questions at the poster session. Thank you.